Hello again, class. I am back again with objectives 3 and 4. So I'm going to get right to it and share my screen. Let's see here. Share. All right, let me get that slideshow up there. Okay, well, today we are going to describe the structure and function of the blood-brain barrier. So we're on chapter 1, objective 3. So what is the blood-brain barrier? Well, it's exactly as we say it is. It is the barrier in between the blood and the brain. Well, the blood is great because it gives us glucose and oxygen and all that, and the brain needs that, but we don't want a lot of other things that are can be in the blood sometimes, like bacteria and viruses. So what we have around the blood vessels going into the brain is a very tightly packed cells, and I'll show you those on the next screen. They're called endothelial cells, and they keep most viruses and bacteria out of the brain, but some stuff does have to pass through, like oxygen, and oxygen passes through without a problem. It just diffuses right through. When we do get a virus into the brain, as I mentioned in the last one, we've got this microglia that is kind of the brain's immune system, but it doesn't work all that well. That's the problem. So we can't a lot of times get those viruses out of the brain. So some viruses, like for example, the herpes virus, will stay with us forever, and it might end up giving you shingles later in life, or from herpes on the lip, or genital herpes, that it keeps coming back and it keeps coming back, and that's because it resides in the central nervous system. And once you get it up into the nervous system, it's really hard to get it out. So a lot of drugs and psychoactive drugs, like for example, heroin or nicotine or alcohol, they cross very easily. Many of these are fat soluble. So those endothelial cells are kind of fatty, and they just go diffuse right through the fat without a problem. Okay, so here's a blood vessel in the brain, and you can see here that there's going to be blood in the middle of it, and that fat soluble molecules will just pass right through. We also have what we call active transport for some things like glucose and amino acids and stuff like that. But most things basically stay out. We don't want them to get into the brain because a lot of viruses and bacteria can damage your brain. So as I said, there's this system called active transport, and what it does is actively pumps chemicals into the brain. And so it'll pump them across that blood-brain barrier. And so glucose is one of those chemicals. It needs to get into the brain so that we are always pumping it in. We also require oxygen and a vitamin called B1 or thiamine to use that glucose. One of the things that happens to some people with what's called Korsakoff syndrome is somebody who's an alcoholic who basically has stopped eating and is just drinking alcohol. They will tend to lack the vitamin B1, which we mostly get from food. And over time, that we're going to see when we get to memory, it causes some pretty significant brain damage and memory problems. But of course, you have to be an alcoholic for many, many years and not eat generally, because you're getting all your calories from alcohol, basically. Okay, objective 4, we're going to describe the nerve impulse and how it all starts with the resting potential. So mostly I'm going to be describing here the resting potential. And so here, let's take a look at what's happening first with the neural communication, because I think you'll get a sense of what happens with neural communication. If you look at the neuron here, again, we're going to get information coming in from dendrites, coming down through the axon and sending out this way. Well, what we have is basically electrical conduction coming down the axon, but what's coming in the dendrites is actually chemical. So here's 2 neurons. In between them, there's a little space. There's a chemical transmission that happens there in between the neurons, but the neurons are mostly going to be involved in electrical conduction. So anyway, we'll talk about that a little bit later. Okay, well, let's take a look at the resting potential. Well, the resting potential basically happens when the neuron is essentially doing nothing at all. And with no stimulation inside the neuron, we're going to then see what's called this resting potential. So the neuron with no stimulation, the inside of it is negative with respect to the outside. So within the cell body, within the dendrites, within the axon, all of that has a negative charge. We say that that's polarized because the inside is different from the outside, just like North Pole, South Pole. And it's about, the charge is about negative 70 millivolts, around there. And so we've got some charged particles, or what we call ions, that are more concentrated outside and some that are more concentrated inside. So for example, we have one called sodium, and that is designated with the Na+. It is a positively charged particle or ion, and it is more concentrated in the outside of the neuron. And so you can think of that as, nah, you can't come in, because it's outside and it's not sort of allowed to come in. Chloride is the same way. Chloride is a Cl-, and you can see there it has a negative charge. It is also more concentrated on the outside of the neuron than on the inside of the neuron. And so I think I can remember that because chloride clashes, so the neuron doesn't really like it. Okay. And then we have a positively charged ion that's more concentrated on the inside, and that is called potassium. It's designated with a K. I know that makes no sense, but it's K+. And I think of the K as we're going to keep it. So we keep it inside the neuron. And there are also some negative proteins that usually are designated with that A- there, and those are going to always stay inside the neuron. So if you look at this little table here in the bottom, you can see that potassium, there's some of it outside the neuron, but most of it is inside. And most of the chloride and most of the sodium is also outside of the neuron, not inside. So you got to kind of remember what's inside and what's outside, and then we'll talk about how these things move around. But at any rate, all the combination of pluses and minuses make it about negative 70 millivolts inside when the neuron is not doing anything. Okay, well, these ions, potassium and sodium and chloride, are all affected by two forces. One is the concentration gradient, and one is the electrical gradient. The concentration gradient basically says that molecules are going to evenly distribute throughout a medium. So for example, if you take a thing of water and you put some food coloring in it, what you find is that the food coloring looks very concentrated at first on the top, and then it just distributes throughout the water and everything becomes a pretty even color. So molecules will tend to kind of spread out. And so what happens to ions is the same thing. If you have a lot of sodium in one place, it will tend to want to spread out to other places and kind of evenly distribute. The electrical gradient is one you're probably familiar with by working with magnets or whatever. The electrical gradient is just that positives are attracted to negatives. So if you have a negative 70 millivolts inside the neuron, you can imagine that you have some sodium out here. Remember, the sodium is a positively charged ion. It's going to want to come inside the neuron, right? And so you've got some things inside, some things outside. And so we kind of have to look at where do things want to go? Well, we've seen sodium wants to go inside because it's positive and the neuron inside is negative. It's also more concentrated on the outside, so it wants to go inside because it wants to move to an area of less concentration. That's the concentration gradient. If you take chloride, you can see that chloride is negative. Now, the inside of the neuron is also negative, so is it going to want to go in there? No, it doesn't want to go in there because it's attracted to positive, not negative, by the electrical gradient. By the concentration gradient, however, it does want to go in there because it's more concentrated on the outside. So you can see chloride remains about the same, and potassium will do the same. Potassium is going to remain about the same because remember, it's more concentrated inside the neuron, so it's going to want to go out by the concentration gradient, but it's positive. So it likes that negative 70 millivolts inside the neuron. It wants to stay there because of that. So it wants to go out by concentration, wants to come in by electrical. So you can see now sodium is quite different because sodium is going to be drawn in by both forces, whereas the other ones are a little bit more stable because one force kicks them out and one force draws them in. Okay, so all of these things have to pass through these protein gates to get in or out. And when the neuron is at rest, when it's in resting potential, the gates to sodium are closed, okay? So sodium can't get in, okay, at all, okay? Potassium wants to get out by the concentration gradient, but it's held in by the electrical gradient because it wants to be in that negative 70 environment because it's positive. Chloride wants out because it doesn't like that negative environment, but it's held in by the concentration gradient because there's a lot of concentration of chloride outside, and so the concentration gradient would want to draw it in. Okay, there is something that we call the sodium-potassium pump. And what the sodium-potassium pump does is it draws in 2 potassiums for every 3 sodiums it pushes out. So it's always pushing out sodium, more sodium, and drawing in more potassium. So that's why we have a lot of potassium inside. We have a lot of sodium outside. Now, you can imagine that if you open up the gates for sodium, then sodium is going to rush in, because remember, sodium has an electrical gradient that wants to draw it in, and it has a concentration gradient that simultaneously wants to draw it in. So if you open those gates, the sodium is going to rush in. And so in some sense, the resting potential is sort of like a cocked gun, because it's sort of ready to go off, because as soon as those sodium gates are opened, what we're going to see is sodium rushing in by the ton. Okay. So I want you to think, can you explain this? What is the resting potential? What are the constants? How do the concentration and electrical gradients affect each one of these? And I want you to be able to explain that. And then what's the point of the sodium-potassium pump? So let's just go through these. The resting potential is when it's negative 70 inside. I want you to know those electrical gradients are positives attracted to negatives, so go through each one of those that way. And the concentration gradient means if it's concentrated in one place, it wants to go to an area of less concentration. And then the point of the sodium-potassium pump is to kick sodium the hell out and get that potassium in. Okay, and we'll work more on Objective 5 in the next video. You guys have a great day.